Today's case study connects plant transport tissues to a real-world situation you may have seen online or in flower shops: rainbow-colored roses. As we work through this scenario, you'll think how water moves through a plant and how dissolved substances can travel along with it. Michael wants a colorful, meaningful gift, but has a limited budget. Instead of buying already dyed flowers, his aunt suggests a do-it-yourself approach using white roses and food coloring. Before we talk about how this works, think about what normally moves through a flower stem and how that movement might be used to carry color. Plants constantly move water from their roots to their stems, leaves, and flowers. Any substance dissolved in that water can potentially move along with it. In your lab, you identified the tissue responsible for this upward movement. Keep that tissue in mind as you think through this case. Pause the video and write your response to reflection question 1 in your lab notebook. Think about what part of the rose would be placed in the colored solution and how the dye might move from that point to the petals. If water normally moves from the base of the stem to the flower, what would happen if that water contained a colored substance? What path would it follow inside the stem? When you resume, we'll move on to the next question. Pause again to answer reflection question 2 in your lab notebook. Think back to your herbaceous stem and vascular tissue observations. Which tissue moves water and dissolved minerals upward, and what specialized cells does it contain? Pause and write your response to reflection question 3. Think about what characteristics a plant would need for this to work. Maybe open water conducting tissue, actively transpiring petals, and light-colored tissues that would show dye clearly? Ask yourself, would woody stems behave the same as herbaceous stems? Would dark-colored petals show dye as easily as white ones? Would plants with blocked or damaged vascular tissue transport dye effectively? In this case study, you connected plant anatomy to a real-world example by considering how food coloring could move through a rose to create rainbow-colored petals. You reviewed how water is transported upward through the stem and how any substance dissolved in that water can travel along the same pathway. The appearance of color in the petals makes this internal transport system visible and helps you visualize how vascular tissues function in living plants. This example reinforces the idea that plant structure supports function. The same tissues that normally carry water and minerals can also transport dyes, allowing us to observe movement that would otherwise be invisible. In this lab, you explored how plant structure is organized from the cellular level to the level of whole organs, focusing on tissues and stems. You began by examining simple tissues. Parenchyma cells function in storage and metabolism. Collenchyma provides flexible support in growing regions, and sclerenchyma provides rigid support through thick lignified walls. Next, you studied dermal tissues. The epidermis protects young non-woody organs and regulates gas exchange through stomata, while the periderm, including cork, replaces the epidermis in woody tissues and forms a waterproof protective barrier. You then investigated vascular tissues. Xylem transports water and minerals and provides structural support, while phloem transports sugars and other organic nutrients. The position and structure of these tissues within vascular bundles allowed you to distinguish them microscopically. By comparing herbaceous monocot and eudicot stems, you observed how vascular bundles are scattered in monocots and arranged in a ring in eudicots, and how cortex and pith function as crown tissues. This highlighted how internal organization relates to growth patterns and support. Finally, you examined meristematic tissues at the stem tip and saw how apical meristems, along with protoderm, ground meristem, and procambium, give rise to all primary tissues and drive stem elongation. Overall, this lab demonstrated a central theme in plant biology: structure supports function. The arrangement and specialization of cells and tissues within stems allow plants to grow, transport materials, remain upright, and interact with their environment.